Hi everyone, this is our project, Sex Differences in VGLU2 Expression in the Locus Coeruleus in relation to Parkinson's disease. I'll be starting off by talking about the background. So, an important theme in our class was the recognition that sex differential research in neuroscience was not as prevalent as one might assume, even though morphologically males and females are very, very different. This reminded us of Parkinson's disease, as it is a neurological disease that presents in males up to 2 times as much as it does in females. Upon further literature review, we discovered that neurons in the locus coeruleus, or the LC, degenerate much before even the hallmark dopamine neurons in Parkinson's do. In addition, VGLU2, which was our chosen gene or protein of interest, was found to be neuroprotective, meaning that it aids in striatal innervation, meaning that essentially it helps mitigate damage done to basal ganglia regions in neurodegenerative disorders. However, VGLU2 is also upregulated in Parkinson's disease models. Furthermore, norepinephrine neurons have been shown to commonly express VGLU2. Because a large majority of LC neurons express VGLU2, and because of a relative lack of research investigating VGLU2 in norepinephrine populations in the brain, we were intrigued. Given this information, we hypothesized that females would have higher VGLU2 expression in the locus coeruleus compared to males, on the basis that VGLU2 was neuroprotective, and females suffered from Parkinson's disease less so than males did. This research is important because it highlights differences in the ways in which male and female brains function, contributing to a growing amount of literature focused on reducing outright sex omission from methods and reducing sex bias.
So for our experiment, we chose to perform immunohistochemistry, and for this we used 5 male and 5 female mice brains that were perfused and given to us by the Developmental Neurobiology Group at the National Institute of Environmental Health Sciences. The brains were then placed in tissue freezing medium prior to cryosectioning of the hindbrain that we did in class. We then incubated our cryosectioned brain samples in our dual primary antibody solution and our dual secondary antibody solution, both of which were used to visualize and quantify the VGLU2 expression and our norepinephrine neurons. The primary antibody solution was used to confirm the presence of VGLU2, which can be seen in the red in the figures following, while the secondary antibody solution was used to visualize GFP, which can be seen as the green, and was used to label our norepinephrine neurons. Our brain slices were then organized by neuroanatomy, and then we mounted them onto slides. Following that, we used a Nikon light microscope along with NIS element software to visualize our neurons and obtain images for further analysis. We also used ImageJ and Fiji software in order to quantify VGLU2 expression and our norepinephrine neurons. We first auto-adjusted the images that we acquired for brightness and contrast, and then estimated background intensity from nearby regions that did not contain fluorescence, and then subtracted it to calculate background corrected fluorescence, which was then normalized by a region of interest area to ensure optimal quantification of VGLU2 expression. We then calculated this per area and per the norepinephrine nucleuses, and then compared that between male and female mice. We then used GraphPad Prism, which is a software used for statistical analysis and to create our graphs that can be seen in our results following.
Yeah, so the Allen Brain Atlas provided us some preliminary evidence that VGLU2 is expressed in the LC because it's circled in the black box. You can see that DBH norepinephrine neurons, and then compared to VGLU2 expression, and VGLU2 is expressed in the LC. Going to figure 3, we first showed that in the LC, VGLU2 expression was significantly above the negative control, which excluded the primary antibody for VGLU2, and that is indicated in figure 3A, where there is no red. And then figure 4 shows the differential VGLU2 expression in the different norepinephrine subregions. So the LC had the greatest mean expression of all norepinephrine subregions and was significantly different from the A1, A5, and A7. But it was close to the average expression in the sub-CD, which was interesting. Looking at the general sex differences in VGLUT2 between males and females, we see a similar expression of VGLUT2, which could be because VGLUT2 expression is like ev evolutionary, evolution, well, conserved and um, important for life. And so, since we saw no significance there, we looked into um, this subregion, the LC subregion, and found a significant difference between males and females, um, with males having higher VGLUT2 expression than females, contrary to our hypothesis, which might mean there's some functional significance there that future studies could look at.
To summarize the results of our experiment, we confirmed that the Locus coeruleus had the greatest average expression of VGLUT2 than compared to any other subpopulation. And additionally, we found that male mice expressed significantly greater VGLUT2 expression than females, which was contrary to our hypothesis, and which could suggest an alternative explanation, potentially involving estrogen, to explain the sex difference. Some major limitations for our experiment is the small sample size that we have to work with and a lack of a Parkinson's disease model. I think in the future, if we improved and upregulated the sample size and if you had an actual Parkinson's disease model, it'd probably validate our results more. Some future research which we could focus on would be looking at R1 rhombomer fate maps, since they have high VGLUT2 expression and are abundant in the LC and sub-CD. Additionally, it would be interesting to look at the estrogen's neuroprotective effects in Parkinson's disease, because previous research has shown that estrogen suppresses inflammatory reactions and protects nigrostriatal dopaminergic neurons by regulating gene expression. So this could be a potential link between estrogen and Parkinson's disease. This research is important because it provides an avenue for sex-specific medications for patients with Parkinson's disease and similar neurodegenerative disorders. And that is the end of our poster. So thank you for watching.